Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
Good afternoon, everyone. My name is Daniela Egan, and I am the Director of Class Giving in the MIT Annual Fund. On behalf of the MIT Alumni Association, it is my pleasure to welcome you to today's program, a talk by Professor Michael Birnbaum. Once Professor Birnbaum begins, you can feel free to add questions in the Q&A feature at the bottom of your screen. To introduce you to the work of the Koch Institute and Professor Birnbaum, we would like to start off by playing a short video narrated by Erica Reinfeld. Enjoy.
Welcome, everybody. My name is Erica Reinfeld. I run the outreach program at the Koch Institute. Normally, I'd be welcoming you into our auditorium and offering you a tour of our public galleries seen here. But in light of the current situation, you are just going to have to make do with my green screen and a rather whirlwind overview of what happens here in Building 76. So before there was Building 76, there was E19. And before it was E19, it was an old candy factory. The very cleverly named MIT Center for Cancer Research was founded in 1974 and designated by the National Cancer Institute as a basic cancer research center. And it supported a wide range of cancer-relevant science, including molecular biology, genetics, and immunology. Then about 10 years ago, still with our NCI designation, we moved across the street to become the Koch Institute for Integrative Cancer Research, both improving our physical spaces and more than doubling our workforce by recruiting cancer-oriented engineers to join our faculty. By bringing biologists and engineers together under the same roof, our goal is to rapidly accelerate both discovery and innovation using interdisciplinary approaches. So this is our mission: to develop new insights into cancer, as well as new tools and technologies to better detect, monitor, and treat the disease. And we've also identified 5 research focus areas that we believe are critical for rapid progress against cancer. I don't have time to share my favorite projects with you. Of course, I don't play favorites, but in short, those focus areas are precision cancer medicine. How do we find the right treatment for the right patient at the right time? Cancer nanomedicine, using nanotechnology, very, very tiny technologies, to improve drug delivery and a whole host of other things as well. We want to understand metastasis, which of course is the spread of cancer cells through the body, and as too many people know, the leading cause of cancer mortality. We're also very interested in detection and monitoring, how we can find cancer early and then track its progression as it develops or responds to treatment. And then finally, cancer immunology, understanding the interplay between tumors and our body's natural defense system, and then engineering that immune system to better fight cancer. And trust me, Michael can tell you a lot more about that one in particular. Because these focus areas are so cross-cutting, we're able to apply the approaches and the technologies across many different cancer types. And then, of course, this interdisciplinary collaboration among ideas and people is also applicable to other biomedical challenges as well, which is why so many of our researchers have been able to pivot existing research to address things like COVID-19, for example, and contribute to global health solutions more broadly. So before I turn you over to Michael, I did want to highlight 2 of our signature programs, which are essential for making sure that the cancer research we do helps those who need it the most. Both of these programs are philanthropically funded and absolutely crucial for that bench-to-bedside translation. The Bridge Project is a collaboration between the Koch Institute and Dana-Farber Harvard Cancer Center, which is Boston's other NCI-designated cancer research center. And it brings together MIT researchers and clinical oncologists to jointly tackle the most intractable problems in cancer research and care. And then the Frontier Research Program is the other end of that pipeline, providing seed funding for early-stage projects that might not qualify for traditional funding. These are high-risk, high-reward projects, things like a urine test for early cancer detection or origami-inspired drug delivery, and they're funded through grassroots efforts. So I would absolutely love to tell you more about all the examples on this slide, but I know you're actually here to learn from Michael, so I will just encourage you to visit our website or subscribe to our newsletter to learn more about the collaborative interdisciplinary research that we do. So on that note, it is my great pleasure to introduce you to Professor Michael Birnbaum. Professor Birnbaum is the Eugene Bell Career Development Professor in Tissue Engineering and an Assistant Professor of Biological Engineering. He holds degrees from both Harvard and Stanford, but we are nonetheless thrilled to have him as part of the MIT community. His laboratory studies immune receptor recognition and function in cancer, autoimmunity, and infectious disease to understand and then manipulate immune responses. So in other words, in addition to teaching MIT students, Professor Birnbaum teaches the immune system how to better overcome disease. I hope you enjoy his talk and look forward to seeing you on campus in the future.
Great. Hi, everybody. I'm Michael Birnbaum. Erica, thank you for that introduction, and thank all of you for joining us today. I'm very excited to be able to present some of the things we have going on in my lab to you, and hope to be able to do it, as Erica said, in person as soon as we can. So as a bit of background, I'm an assistant professor in the Department of Biological Engineering at MIT. And I'm actually an immunologist by training and an engineer by vocation, which is actually a fairly common occurrence at MIT and in the Koch. And so the work that I'm going to be telling you about today is some of our efforts to try to mate immunology and various types of bioengineering, in particular protein engineering, in an attempt to better understand and manipulate the immune system. So I'll be telling you about that, some of the things that we're doing both for cancer and for infectious disease, most recently COVID-19. And in the meantime, just in terms of logistics, if you have questions as we go, if you put them into the Q&A box on your Zoom interface, we will have plenty of time to answer some at the very end. So with that, let's get started. So I figured I would start by making you all experts in about 2 slides on how the immune system works. So when your body encounters some type of protein or pattern that could be dangerous to it, so broadly called antigens, and these might be viruses or bacteria, they could be parasites or fungi, they could be a tumor or a transformed cell when the body is working correctly. It could be a cell that you wouldn't want recognized when the immune system is working incorrectly and causing something like autoimmunity or a transplant rejection. Whatever these sources of antigens might be, they're taken up by specialized cells called antigen-presenting cells, and then broken down into small peptide antigens. So these are just short amino acid sequences that come from the protein. And then they are presented by specialized molecules that are on basically every cell of your body called MHC molecules or HLA molecules. So these are the molecules, they're quite diverse. Mine are very likely different than any of yours, unless my parents happen to be on this seminar, and are the basis of, for example, what makes organ transplantation difficult to find matches for. But these combinations of peptides bound to MHC molecules, I'll call them peptide-MHCs, in my presentation to you today, are then recognized by a key player in the immune system called a T cell. And T cells, although it's not going to play too much in today, have a couple of different jobs, but each of their jobs rely upon exquisite recognition of only particular peptide-MHC antigens in order to then turn on and initiate an immune response. So there are helper T cells whose job is mainly to coordinate the rest of the immune system. So they're needed in order to make effective antibodies. They're needed to make sure that all of your macrophages and other cells are doing all of their jobs. And then there's also cytotoxic T cells, which are the ones that are given the power to actually kill other cells when necessary in order to combat disease. And so this is an area that I've been interested in really ever since I learned about it as an undergraduate. And one of the things that has me interested and sort of ties into how I can become an engineer too is the numbers problem of the adaptive immune system. So like I said, the peptide bound to an MHC molecule is the key feature that's recognized in order to be able to tell whether a cell has something wrong with it, whether there's a viral infection or a bacterial infection or cancer. And T cells use a specialized molecule called a T cell receptor in order to recognize this pattern. And pretty uniquely in the body, it's only T cell receptors and antibodies. These are actually recombined or made uniquely in every T cell. So every T cell has its own T cell receptor. And the math of this begins to get very large. So each T cell receptor, there's a estimated 10 to the 15 possible ways that you could make one, which is a very large number. In you or I, we have about 10 to the 11 T cells that are in each of us. And maybe 10 to the 8 that are unique because they expand, and so you have many copies of any individual T cell. Our mouse models have a smaller number that's commensurate to the fact that mice are smaller than us. But if you just do the math for how many different patterns, how many different MHC molecule peptide combinations are possible to be seen, that ends up on a number that's somewhere around 10 to the 12, so about 1 trillion possible combinations, a very large number indeed. So how could this work? Because one thing that seemingly can't happen is one T cell per possible antigen, because this has been looked at before. And if you look at the number of possibilities, if it were 1-to-1, for example, you would need a box of T cells 70 times the size of this box relative to this mouse in order to cover this mouse. So that's not likely. But we also know that to a first approximation, if there's an antigen that's out there to be recognized, you can have a T cell that recognizes it. So with those two things in mind, the immune system has to be incredibly cross-reactive, on the order of every single T cell has to be responsible for on the order of 100,000 different peptide-MHC antigens that are there. Not only is it cross-reactive, but it also has to be incredibly sensitive. So this is actually data from about 20 years ago from one of our other Koch Institute faculty colleagues, Daryl Irvine, showing that the system can recognize even just a very small handful of antigens. And these are at very, very low affinities as well. So for those of you who aren't biologists or chemists, what I could tell you is this is the equivalent of things coming together and apart, kind of like this, on the order of staying together for less than a second. And a T cell is going to be able to recognize just a handful of those and then make a literal life or death decision about the cell that happens to have the antigen on its surface. So the system is incredibly sensitive, cross-reactive, and yet has to be the basis of whether or not we have an effective immune response protecting us from cancer or infectious disease, or whether it's something that could inadvertently initiate some type of autoimmunity. And then less important for today's talk, but it's also plastic. And what I mean by that is that the signal through the T cell receptor into the T cell can actually lead to a great many different factors of what the T cell actually does with that information. So this is an area that's both very interesting for us in terms of just the theory of the system, but of course has very practical implications as well. Some of them for infectious disease and vaccination are self-evident just in terms of day-to-day life, particularly right now. But this also has become increasingly true for cancer. So in the last 15 years or so, there's been a true revolution in how we think about treating tumors and cancers through immunotherapy, which is basically repurposing or unleashing the potential of the immune system in order to be able to target a tumor. And many, if not all, but many of these treatments involve a T cell one way or the other. And this goes anywhere from vaccinating against the tumor, much in the same way that you would vaccinate against the virus, to unleashing the inherent potential of a T cell that might be in your body, tried to eliminate the tumor and couldn't, so basically giving it a boost or turning it back on, all the way through to engineering the T cells themselves in order to give them new function and new specificity, take the cells out, do something to them and put them back into a person. And the reason we're also excited about this, one of the reasons that I gained the privilege of working in the Koch Institute, was because of trials in people that look something like this, where these are examples of people who have highly metastatic melanoma, where when they're given a treatment called checkpoint blockade, which is one of them that basically turns T cells back on that otherwise were turned off for whatever reason, in a sizable number of these patients. And these are patients who 20 years ago would have likely succumbed to disease in a fairly short time frame, actually have a very long-term survival. So, we're always very careful about using words like cures, but these are people who survive for much longer out in terms of months and even years than they would otherwise. But something that we're still working out is that this is true for some cancers, but not all cancers. So melanoma is a case where this works well. Lung cancer is a case that works well, but it's not all types of cancers. And then even in cases where it works well, it's some people, but not all people. So you can see here that in the case of this trial, it was on the order of maybe 30 or 40% of people who had a good response, which meant that 50 or 60% of people didn't and succumbed to disease. So we want to better understand how that works. We also know that these treatments still carry fairly serious side effects. So in some cases, what could be seen is that when you're unleashing the potential of T cells, you're not just turning on the T cells that recognize the tumor, but you're also turning on T cells that recognize the rest of your body. So people who had cancer would have their cancer cured miraculously, but also then end up with inadvertently diabetes, for example. So we want to be able to predict why that is and what we could be doing better. And if we could, instead of unleashing the entire immune system, try to just target T cells in the way that you would target an infection for a vaccine. So there's your whirlwind tour of immunology. But just as we have that in mind, that's where my lab is thinking. And so in my lab, we work almost entirely on T cell recognition and T cell signaling. And so the types of questions we're interested in are, what do T cells recognize during the course of a disease? And how could we potentially better manipulate that? So how can we induce better responses? This is true for both cancer and infection. And then we also want to know, can we better engineer T cell signaling as well to create better responses than what nature could naturally do? And so I'll, for the sake of time, we'll be focusing on the left half of this, but if anybody has questions on the right half, feel free to reach out to me after. So like I said, one of the promising, more recent approaches for therapy is to try to treat cancer and cancer immunotherapy in the same way that we would treat an infection and try to vaccinate you against your own tumor. And advances in really sequencing technology, DNA sequencing, has made it feasible to essentially take a sample from a tumor, take a sample of your normal body tissues, sequence them in order to find the places where the tumor has mutated, and then try to induce an immune response, train your immune system to target just those places where there's differences, with the idea being that those might be the more safe places to target because by definition, those are going to exist in the tumor and not elsewhere. So these are so-called neoantigens. And this has been done, this entire process, from sequencing through to vaccination. And there's a few companies who are now trying this. This has been, this paper I'm showing you here was from an academic lab at Dana-Farber and Kathy Wu's lab, who's across the street from us. And these are patients who had good responses. But in looking at what are the types of issues that are going to keep us from having this be a more widespread approach to do this more broadly, it's actually a step that's sort of on this pipeline here, which is perform epitope prediction. So that's the step where we're trying to figure out from these mutations, which one of them are likely to make for good antigens. And one of the first things that you have to do to be a good antigen is actually bind to an MHC molecule. And not only that, but to go into the weeds just a little bit more, it has to bind to a class 2 MHC molecule, which is the one that the helper T cells recognize for some of the effect. And so what I'm showing you here on the right of my slide is how good we are at being able to do some of these predictions. And we've gotten better over time. So this slide's a little old, so probably we've done better. But if this were perfect, you would have the equivalent of like a right angle triangle right here. But that's not what we end up. This is adequate, but not perfect. And not only that, but it's unequally adequate. So if there are very highly studied MHCs or highly prevalent ones, then we do better than on ones that are more rare. So for example, I have more rare ones just by chance. And so the prediction would work less well for me than it might work for some of you. And so why are there differences in those predictions? Well, part of it is just the amount of data that we have. So if we just look for all of the examples of peptides that we know could bind to MHCs, we have a few 100,000 examples in the literature, which seems like a fairly big number, but keeping in mind that the MHCs are diverse, if you actually break it down for each MHC molecule, for some highly studied ones like HLA-A2, which is an extremely prevalent molecule, particularly in Caucasian populations, there's on the order of 20,000 peptides that are known. Where for some of the more rare alleles, so again, so for example, HLA-DR402, that's me, there's only a couple hundred that are known. So there's a need to get more data in order to figure out what a good example looks like versus what a poor example looks like in order to essentially train algorithms better. So this is where we come in my lab. We spend a lot of time thinking about how to build tools to generate big data sets to better understand how the immune system works. And so one of them that we used, actually developed during my training at Stanford, is something called peptide-MHC yeast display. And so this is an extension of a technology that was first developed by Dane Wittrup, who's another Koch Institute engineer and faculty member. And what we basically are able to do is use microbes like yeast to create very large libraries of different peptides in order to find the ones that have a feature that we want. So here, we're creating libraries that are on the order of 10 to the 8, so on the order of 100 million peptides, and then asking which one of these peptides can bind to an MHC molecule. And then of those, go through iterated rounds, find the ones that can bind tightly, and then sequence the ones that were able to make it through the end of our process, so via next generation sequencing. So we could end up with potentially very large data sets in order to understand what could bind to a particular molecule. So we've gone through and we've done this. So we did HLA-DR4 as a model system. So that's one that's actually fairly well studied to begin with. And this is what the data looks like. And so I'll take you through if you're not used to looking at plots like this, totally understandable. This is our motif. So I told you the peptide is a relatively short stretch of amino acids. So really the most important stretch are these 9 amino acids. And some of the side chains for the amino acids point in and interact with the MHC, some point up for the T cell to see. So when you see these big letters, that is a constraint that's being put on by the MHC. And so we find that there are certain positions the MHC cares about, and each position has a unique amino acid pattern, like a fingerprint, that would be recognized. So this is what we find with our data. If we look at, so this here, Sifpethi, I don't know who came up with that name, but this is a curated data set of peptides that should be good. And so you can see that we match this quite well. But if you look at some of the older algorithms, what they would predict, this is the basis of what you would call an adequate prediction. So you can see that it matches some of the patterns. So it gets this first position pretty okay. The exact rank ordering here is probably not that big of a deal. But it misses even the importance of certain positions, and it misses the importance of particular residues as well. And then another way of looking at it is just like a heat map. We don't have to really go into that, but you get the same type of fingerprinting pattern, just put in a little bit more of a quantitative way. And note that we end up with on the order of 95,000 peptides from this experiment, which is expands upon the about 5,000 that are in the databases right now. So in a single experiment, we could get on the order of 20-fold more data than you would be able to get otherwise. So we did some testing of this, right? So what do these differences actually mean in terms of predictions? So you could imagine that you could be both missing things that should bind that an algorithm would tell you won't, and then there would be things that that don't bind and an algorithm said should. And so we've tested both of these. And so here I'm showing you a whole bunch of peptides that by kind of one of the standard predictors or a couple of the standard predictors would say they shouldn't bind at all. And so we went and actually physically tested them. And what we found was that for basically all of the ones that we tested, if the algorithm said that they should not bind, if our data suggests that they should, when we actually go through and do the test, we found out that they do in fact bind. In fact, close or sometimes even higher affinity compared to a peptide from flu that's known to be a good bind. When we went the other way, we identified a bunch of peptides that the algorithms say should be good binders. So this happens to be from influenza. So these are epitopes that were predicted to be good binders and therefore might be the type of thing that you would train a vaccine to go after. Our approach said probably wouldn't work. And when we actually tested them, they don't bind at all. So had you designed a vaccine against these, that probably wouldn't have worked all that well. So another way to quantitate that would be looking at data in a couple of different ways. For the sake of time, really you want to look here, because not only do you want to know if something binds well versus binds poorly, you want to know, or sorry, not just does it bind or not, but you want to know whether it binds well or if it binds poorly. And so when we look at data like that, this is for us here in the bottom left, a prediction of strong binders versus weak binders, and then what the actual measurement is. So we're not perfect, but we do a much, much better job with our approach, figuring out not just do you bind or not, but how well do you bind. So for example, neon MHC2 is another state-of-the-art technique that actually does quite well in terms of saying, do you bind, yes, no, but does quite poorly for saying how good of a binder are you. And so we're pretty excited about these as results. We're of course keen to adapt this for COVID-19 work as well. And so we did similar predictions for SARS-CoV-2. And what we found was that there are a set of peptides that the current approach would have missed, and a set of peptides that the current approach say probably work that we disagree and think probably would not work. And so we're working with collaborators to figure out, which is true and which is not, which is going to be important to know for understanding what's recognized during the course of a natural immune response against the coronavirus, and also what we should be directing things against. And the answer is likely somewhere in between, right? Like we're probably not getting everything right. We're also probably getting some things right. And so we're going to be working within the larger scientific community to figure out what that might be. And then moving forward, we're working very closely with computer scientists at MIT to figure out, okay, how much does data matter versus how much does the computational algorithms that are trained by the data matter? We're working to directly assess specific libraries for viruses or bacteria. And then it's very important to me to try to do this in a globally oriented way, to study the molecules that could help everybody around the world, rather than the couple of molecules that might be most highly represented in the developed world. And so we're working to do what we did for a couple of molecules for another 40, which we think should cover on the order of 95% of the global population. So what else could we do with this? Well, we can also find a footprint, a so-called footprint of not just what peptide will bind to an MHC, but what the T cell receptor could actually see. So what is the source of response? This is actually where some of this work started when I was a trainee at Stanford. And so this is actually what we built the technique to do at first. And so an example of what that could be good for is we have Australian collaborators where they have identified T cells that during the course of a CMV infection, which is a very normal chronic disease that actually just by chance, something like 80% of you probably have right now, and you just live with like normal. And so there are T cells that recognize peptides from CMV that are important for the antiviral response, that actually then they're doing a good job recognizing the virally infected cells, but then also inadvertently cross-react with your own healthy cells. And it's known that it recognizes HLA-DR4, but it's not known what peptide causes this. So it's a place where a viral infection can inadvertently cause something that looks like an autoimmune disease. Yeah, there we go. So we wanted to know, okay, what peptides might be causing that? What peptides could be, what would their motifs look like? What proteins might they be coming from? So we did our technique again, took a look at what that footprint would be. So now here, it ends up with a very set footprint because it has to bind to the MHC. So that sets, for example, here, here, and here. But it also has to bind to the T cell receptor, which sets the rest of it. So we're able to find a pattern which wasn't known before in the case of what might be causing this. We can show that those peptides actually do things. And then we're able to mix the experimental with the computational. So here's what that footprint looks like. We're then able to use this to train an algorithm to find other peptides in nature that might look like that as well. And so we're still in the process of doing this. It's the place where we've had to pause the work a little bit just because of work that was underway that had to be ramped down due to the coronavirus. But we actually found that there does seem to be a peptide, actually sort of coincidentally from another MHC molecule that matches this pattern very well. And so we now have a hypothesis of what could be causing disease, what proteins could be inducing this. And we're now working with our collaborators to actually test this out. So these are the types of approaches we're building, but something that we still need to work on in my lab, and something that is much earlier on, but I'm excited to tell you about in my last couple of minutes, is a way to do this on even larger scale. Because everything that I've been showing you is going to be one direction or the other. So for example, if we know that we're interested in one specificity, so maybe one peptide from a virus that we know might be important, and we want to be able to find the T cells that recognize it, well, we have a technology called peptide-MHC tetramers, were developed in Stanford in the 90s, are a way to do this, and they're actually very widely used. If we wanted to go the other direction, we have a T cell that seems like it's important, but we want to know what it's able to recognize in terms of antigens. This has been a harder problem. So this is my yeast display technique that I told you about that we've developed in my lab as a way of doing that. Other people are coming up with ways of doing that as well. But neither of those are the exact problem that we actually want to solve, right? We want to be able to build out the entire map at the same time. We want to know who recognizes who in this. We want to be able to look at a bunch of different antigens and a bunch of different T cells all at the same time. So we've been working hard to try to come up with innovative ways of doing this in my lab. And I'll show you, even though we're not quite there yet, what we've come up with. So what we've done is actually took a look at one of nature's methods of doing things like this, which is a virus. So viruses are, in addition to, of course, a public health concern and fascinating study, a very useful tool for biotechnology. And so what we wanted to know is, so viruses always have to use some type of protein on their surface to get into a cell. What happens if we tried to find a way to basically direct a virus to use whatever protein we wanted on its surface to get into a cell? So we could define how a virus infects different cells. And what if we put peptide-MHCs on the surface of a virus? So they definitely don't go there normally, but could we engineer viruses as tools to be able to do so? The idea being then, we could make a library, a collection of these, mix them with T cells, and then only if you have the right pair between antigen and virus could you infect and get in. And then we could take these out and sequence them. So we would be able to get in one experiment from all of your T cells drawn from you, what those T cells recognize using workflows that are quite familiar to people who are in the field right now, so quite compatible with the current state of the art. So we're still working on this, but we've actually gotten the first and I think hardest step done, which is we've actually been able to reprogram these viruses to use the T cell receptor as a way to get into the cell. So here's a T cell. On the right side of my slide is when you have the wrong antigen. And these are viruses that encode a fluorescent protein to make the T cell make that. And you don't see much at all. When you have the right combination, then you're able to get in and infect. So it's still early days here. We're very excited and working hard on this. But why is this important? Is because we want to be able to characterize large sets of peptides on patient samples, on blood, in high throughput. And to give you an idea of some of the numbers that we need and some of the numbers that we're working towards kind of simultaneously, is if that we had about 100 proteins or 100 peptides, we'd be able to cover all of the predicted antigens from the coronavirus spike protein, which is right now the main focus of vaccines for one HLA molecule. If we had 800 peptides, we'd be able to cover basically all of the predicted peptides, regardless of from spike or anything else, for one molecule. And if we did 4,000, we'd be able to cover all of the antigens that your personal immune system could potentially see. And then these should be fairly straightforward reagents to make for all of the different MHC molecules as well. So we could be able to potentially make something that gets towards personalized and comprehensive immune characterization, which is very much our goal moving forward, and is one that I think is going to be very important for studying the coronavirus. But we also are funded for example, via the KI Bridge program to try to adapt this for metastatic melanoma. We're working with our colleagues at the Reagan Institute to adapt this for HIV. And so I think there's a lot that we could do, and we're very excited about this. And then also, this is a tool that could be used for gene therapies as well. If you have the ability to specifically target T cells, you would be able to then add a gene that you would want to potentially give them a new function in ways that other people haven't been able to do it yet. And so we're still wrapping our heads around what to do with this, but we're very excited about it. So that's the science. And so as I'm thanking people, if you have any questions and answers, feel free to begin typing them. Nothing I will say here probably will lead to more questions. But I did have a lot of people that I wanted to thank. So my lab has been around for a little over 4 years now. It has grown quickly, and this wouldn't be possible without excellent collaborators and excellent sources of funding. So the people who did the work, Garrett and Brooke are 2 grad students who worked on the first portion of what I showed you today, worked very closely in a team. Garrett actually is my first graduate, so he actually had the virtual hooding ceremony yesterday, which was very exciting and a little bit bittersweet that I couldn't be there to actually do it. Connor was the person who's working on the virus work that I showed you at the very end. So this is the lab. We've had a host of funders and collaborators, and then a lot of this work was catalyzed by the Frontier Research Program that, that Erica told you about. So with that, and of course, I'd like to thank all of you for coming and spending your Saturday afternoon with me, and for the Alumni Association for putting this together, and for providing me this cup as well, which is very nice of them. So with that, I'm happy to take any questions that you may have. Great. Okay, so the first batch of questions were sent to me, and apologies in advance if I don't get to all of your questions, but we will go through as many as we can. So the first question, could you put multiple peptides in a vaccine to improve the ability to eradicate cancers? It's a great question. The answer is yes, and this is actually done. So this is how some of these treatments are put in. And so actually, you want to balance between having a lot of peptides in order to get as many chances as you can to have a good response, versus not so many that you overwhelm the immune system. So the way that this is actually done right now is, for example, 20 peptides might be used, and they'll do 5, 5, 5, 5, left arm, left leg, right leg, to literally space out where the immune response would start. But when you have that relatively few guesses, figuring out not just what differences are in the peptides, but which ones you should be using becomes incredibly important. Next question is, can you comment on why the immune system has memory, and why does memory fade and require booster shots? So the immune system has a memory component, which is basically a way to ensure that if you had T cells or B cells that were good for something, that you keep them around for a long time. And in that way, they're already vetted as being important. And so this is what provides protective immunity. This is why vaccines work. And so that's why also generally subsequent exposures to the same pathogen, either you are totally protected, or you have a much worse, or sorry, a much more mild case of whatever the disease is. This is also why vaccines work, because we try to skip the messy bit of actually having a disease, have the vaccine kind of do that for you, and then go right to the memory. So some of these questions are actually still fairly mysterious. So the why of the memory is pretty straightforward, but the why does it fade, and why do different things fade at different rates, is something that we're still trying to figure out as a field. And that's why we have booster shots, to try to get you back up to the level that you were at. Some things like measles provide very long immunity. And so if you have a measles shot when you're a teenager, it will likely last you until very long after. But going after some of the other viruses, so for example, influenza, unfortunately, some of the coronaviruses, that memory is variable. And so subsequent vaccinations are necessary. But exactly where those differences come from, we can observe, but figuring out the why becomes harder. Okay, you talked a little bit about the immune system attacking organs. Does your work look at the systemic inflammatory responses we're seeing in children and adults with COVID-19? It's a great question. The answer is not yet, and it's partially because all of us are learning stuff as we go right now. And so this inflammatory disease is being seen in a relatively small, but still an important subset of young sufferers of COVID-19. Seems to be immune related, but we still need to learn more about the basis of what that inflammatory disease is to understand if it's even one that's going to heavily involve T cells or B cells. So if it is, then we could work with folks who are the pediatricians and the scientists who are working there. But that's still something that still remains to be seen. And so like a lot of things, science is a team sport, and we're going to have to figure out where we could work together. And of course, if there's evidence that that's the case, that there is a T or a B cell involvement, that's something that we would be very important to adapt. Next question, how might your prediction algorithms get used in the clinic? Diagnostic tools or drug development, do algorithms get translated faster than medicines? Yeah, it's a good question. And so, and it's kind of tied in all over. And so one thing is that when we're talking about these types of immune responses, they are inherently personal for a bunch of reasons. Most importantly, your MHCs are different than my MHCs. And so therefore it wouldn't adapt into a drug like, as such, like you would for kind of traditional drug development, which is a bunch of chemists work very hard and you end up with a pill in the end. But figuring out, for example, what these personalized medicines should be for each of us, and then doing assessment or analysis of what those tools should be is something else that's in mind as well. Other questions. Does the system distinguish neutralizing regions of the spike protein? This is a great question, and I should say that I've had a couple of people in my lab who have been working hard on this particular question through the shutdown. So there's been some essential work that's been underway in my lab, at the Koch Institute, in the larger Boston community, that even though the majority of us are shut down, so I've been working from my living room since mid-March, and much of my lab has been home. But there have been some folks who have been working on COVID-19 essential work, and that question is one that's on our minds a lot, and actually on B cells as well. So we've begun a dalliance with B cells along with T cells, and the answer is that we're trying to build tools that are going to be a better match for being able to pick out, in this case, particularly the B cells that would be most effective for neutralization. So that's not work, I didn't have the data in my presentation today, but is work that we're trying to do because the immune system actually gets distracted by making antibodies against pieces that aren't neutralizing. And so being able to sort out the neutralizing pieces from the non-neutralizing pieces is something that we're trying to help collaborators in a few different sites answer that question with our virus technology. Okay, are we going to have a new profession at each doctor practice who designs the vaccine for each patient? It's a great question, and it's something that we're still figuring out as a field as well. So even though a lot of the work that I'm doing is personalized, I'm still actually pretty biased that anything that we could figure out a way to do robustly and not personalized would be the way to do it. And so anytime that you could treat everybody with one drug, so long as it's effective, that would be great. But we also are going to be in a place where the medicine is going to have opportunities to be done in a personalized way. This is a heavy emphasis in the Koch Institute. And part of that is that the biology is going to have to be met with the engineering here, because this means that we have to do a lot more analysis person by person. We have to figure out how to do personalized manufacturing, and all of these things are new to us in the field of biomedicine, and are areas that I think a lot about, and several of my colleagues think about in the Koch Institute as well. Okay, so a couple of other questions, and apologies if we're not able to get to all of the questions, and thank you for asking them. I've just got the warning that we got 5 minutes left. So how does the understanding of genetics inform the studies of immunology? And the answer there is heavily, although it varies disease by disease. So for things like autoimmunity, it matters a ton. For tumor immunology, it matters if you count the genetics of the tumor, it matters a ton. For infectious disease, it matters in some cases. So it matters in HIV in certain ways, where there's a genetic association to those who are more protected from disease. It matters subtly for a lot of other diseases. But a lot of the tools that are developed for genetics are things that are very useful for us. So a couple of other questions here at the end. Is there a test that can be used to, can use these proteins to predict if cancer will come back? So this was asked by a triple negative stage 2B breast cancer survivor. And so the answer there is we don't know yet. And there's an element of why people are excited about the immune system and using immunotherapies is that element of memory, in that you can have the potential for the immune system to be there in order to potentially treat disease. Now, what that means for patients who are cancer survivors with good prognoses in terms of monitoring is an open question, but there are colleagues of mine in the Koch Institute, for example, Sangeeta Bhatia, who are working on ways to create very minimally invasive and sensitive ways in order to have those types of, if not predictions, outright monitoring to be done. Okay, the last question is, with immunotherapies and other therapies, do you think that we'll be able to cure cancer finally? And then asking me to guess on a year. And so this is an important question, and one where it also comes into play that, you know, cancer is a basket of diseases rather than a single disease. And so it's going to be very different for each cancer, and in sometimes not necessarily obvious ways. And so a lot of what people like me are doing right now is trying to match approach with cancer. And this is a linkage between what opportunities are available to us due to the biology, what opportunities are available to us in terms of the biotechnology and the engineering, and then testing it out in medicine. And so, so I think that we're going to see this done excitingly, frustratingly, unfortunately, in a patchwork way, where there's going to be breakthroughs that change cancer by cancer. And hopefully this will end up being synergistic, right? Where we'll be able to take learnings from one place and apply them to another. But it's going to be like we've seen already, I think is going to be what we see. So for example, metastatic melanoma, the prognosis is quite different now than it was 10 years ago, thanks to checkpoint blockade. CAR T therapies, which is an engineered T cell therapy, have really transformed how we think of B cell cancers, particularly in kids. And so these are miraculous things that have happened due to the hard work of a lot of people in certain diseases. And hopefully we're going to be able to keep learning and keep doing this for other cancers as well. But I think that we're still a way away from being able to do this generally. And so hopefully we'll learn more and be able to do the work, but there's certainly a lot of work out there for all of us to do. And so we'll keep busy for as long as it takes, of course. So I think I've used up my time, but thank all of you for coming today. Thank you all for your support of MIT. For those of you who are MIT alums, thank you for checking back in on us. And again, if there's any questions that I wasn't able to answer, do feel free to email me. Take care.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.